Microorganisms are too small to be seen with naked eyes. To study their shape, size, arrangement, and internal structures, scientists use a microscope and various staining techniques.
A microscope is an optical instrument used to magnify very small objects that cannot be seen by naked eyes. It enlarges microorganisms and allows detailed observation of their morphology and cellular structures. The magnification of a compound microscope depends mainly on two lenses. Low-power objective lenses are Mounving nosepiece and oil immersion objective is 100. Total magnification is ocular lens times objective lens. Most microorganisms are colorless and transparent. Without staining, they are difficult to observe. Staining provides contrast and highlights structures. There are different types of staining techniques such as simple staining, negative staining, differential stainings like gram staining, and acid-fast. There are special staining techniques as well, such as endospore staining and capsule staining. Simple staining includes Step 1. Prepare thin smear on clean slide. Step 2. Air dry. Step 3. Heat fix the smear. Observe under oil immersion microscope. Basic dyes bind to bacterial cells. This causes the cells to become colored. Common basic dyes are crystal violet and methylene blue. These are widely used. Bacterial cell surfaces are negatively charged. Positively charged basic dyes are attracted to the cells and stain them directly. In negative staining, the background is stained instead of the cells. Since no heat fixing is required, cell morphology remains undistorted. Acidic dyes possess negatively charged chromophores. Since bacterial surfaces are also negatively charged, the dye is repelled and stains only the background. Differential staining distinguishes bacteria into groups. This is based on differences in cell wall composition. Major types include gram staining and acid-fast staining. Gram staining divides bacteria into gram-positive and gram-negative organisms. Grams iodine, alcohol or acetone are required for gram staining. Prepare and heat fix smear, add crystal violet and wash. Add iodine for 1 minute, decolorize with alcohol, counterstain with safranin. Gram-positive bacteria remain purple because thick peptidoglycan retains crystal violet. Gram-negative bacteria become pink because alcohol removes crystal violet and safranin stains them. Acid-fast staining differentiates bacteria possessing waxy mycolic acid in their cell wall. Carbolfuchsin, acid alcohol, and methylene blue are required. The methodology includes an heat fix, flood with carbolfuchsin, heat gently for 5 minutes, wash and decolorize with acid alcohol, counterstain with methylene and observe under oil immersion. Mycolic acid-rich cell walls retain carbolfuchsin even after acid alcohol treatment. Hence, acid-fast bacteria remain red, while non-acid-fast bacteria become blue. Endospores are highly resistant structures produced by Bacillus and Clostridium species. Examples: Bacillus subtilis and Clostridium botulinum. Prepare smear and heat fix. Cover with malachite green. Steam over boiling water for 5-10 minutes. Wash with water. Counterstain with safranin. Observe microscopically. Heating facilitates penetration of malachite green into resistant spores. After washing, vegetative cells lose green color and become red due to safranin. Capsules are gelatinous protective layers surrounding certain bacteria. Examples: Klebsiella pneumonia and Streptococcus pneumonia. Mix bacterial suspension with India ink. Spread thin smear. Air dry without heat fixation. Counterstain with crystal violet. Observe under microscope. Capsules do not absorb stains. The background becomes dark and the bacterial cell stains purple, leaving a clear halo representing the capsule. Staining techniques are indispensable tools in microbiology. They help visualize microorganisms. Through the microscope, the invisible microbial world becomes visible. I hope the session was interesting. Thank you so much for watching.